Hi students, hope you all are doing well. So, welcome to another exciting session of healthcare entrepreneurship. Today we are going to talk about a very exciting topic which is known as clinical testing methods. So, let us get started. So, majorly while we are working through the different problems and products and services throughout this course, we are specifically looking at a clinical problem, right. So, we start with an idea, we have a design and we finally validate this in either in vitro models or animal models, and we look at clinical testing studies for this as well before we take this out to the market. This may not be required for all sorts of products and services which we offer, but for several of the medical devices or diagnostic products or things which touch with the human body or go inside the human body, they need clinical testing methods, right. So, biomaterials and medical devices constitute an extremely diverse heterogeneous category of items which require clinical testing. Safety of medical devices or material is the primary concern for any inventor, right. So, as an entrepreneur we are also inventors, we are working on a very unique product or idea, like if we are doing this in domain of medical devices or biomaterials which go into any of these devices, we have to look at the clinical testing, right. So, we have to balance between the risk and safety, right. The risks have to be minimal and the safety has to be the utmost for majority of these products. So, the aim is to translate biomedical engineering innovations from lab to the real world applications, right. So, this is only possible by providing the effectiveness or proving the effectiveness in the human subject studies. So, we cannot just prove the effectiveness of an implant or some kind of a material in just few animal models or few in vitro models and say this is going to work fine. This has to prove its effectiveness in humans at the end, right. So, there are several aspects or categories which has to be complied with. The first one is safety. So, under safety we have to look at biocompatibility. I will explain what this term is, right. This is in a generic definition, it is how compatible a particular device or material or an implant is with the human body, that is what biocompatibility means. Surface characters and the operator's safety, right. So, these are some of the important aspects from the safety standpoint. Sterilizability, so it should not be destroyed by autoclaving, dry heat, radiation, ethylene oxide, etc., right. So, these kind of things have to be looked upon while we talk about medical devices. Strength and durability from a manufacturing standpoint or manufacturability standpoint. Strength, durability, machinability, moldability, that are you able to machine it and manufacture it, how much strength is it offering, from the base materials to the actual product which you have built, these kind of things have to be tested as a part of the clinical testing, right, in order to go forth with this product. So, typically what is done is clinical trials. These are done to translate and evaluate the safety, efficacy and performance of the device or material, and also used to optimize and refine. The responsibility lies with the regulators to ensure the safety and efficacy of the medical devices, right. A wide variety of items are classified as medical devices, making medical device regulations a very complex procedure. So, medical device regulation is as we have explained before is into the regulations and standards lectures, like it is an ongoing process where more and more medical devices and technologies are accepted and are regulated. In the initial stages they are not being regulated very well, right. So, it is a very complex procedure, and also what needs to be regulated, is it the material or in certain cases how the efficacy has to be tested, a lot of these things are unanswered, right. So, there is no standardized or universally accepted procedure for medical devices in general, which says that this is the father of all the clinical techniques. So, this is what needs to be followed for all the devices. This changes from case to case basis, right. So, medical device, let us define it very quickly, is an instrument apparatus which basically or implement, machine, contrivance, implant, in vitro reagent or other similar related articles, these all come under the medical devices canvas, which includes any components or part or accessory which is recognized in the official national formulary or the US pharmacopoeia or any supplement to them, right. Like these are typically definition of the medical devices. Any of these aspects which are recognized in the US pharmacopoeia or national formulary or supplement to them, which are intended for the use in the diagnosis of disease or other conditions or in the cure, mitigation, treatment, prevention of the disease in man or other animals, these have to be clinically tested, right. Medical devices are also the products which are intended to affect the structure of or any function of a body of man or animals, right. This is as per the US FDA section 201H definition, right. If it is going to interact or affect any structure or function in man or animals and which does not achieve its primary intended purpose through chemical action within or on the body of the man or other animals, which is not dependent upon being metabolized for the achievement of its primary intended purpose. This is as per the definition, right. This is what medical devices. So, it is a canvas term. Medical devices does not necessarily only mean implants, right. This is a canvas term for a lot of different devices or things which interact with the human body. All devices including an instrument, apparatus, appliance, implant, material or other article, whether used alone or in combination, including a software or accessory, intended by its manufacturer to be used specifically for human beings or animals, which does not achieve the primary intended action in or on human body or animals by any pharmacological or immunological or metabolic means, but which may assist in its intended function by such means for one or more of the specific purposes. This is kind of medical language which is used, but this is the definition for all the devices which are considered to be medical devices. They could be for the purpose of diagnostics, prevention, monitoring, treatment, alleviation of any disease or disorder, right. Diagnosis is a very big segment. So, you may think that how is a medical device specifically for diagnosis? They need not have to go inside the body, sometimes they could be outside the body also for diagnosis, they have to be also clinically tested. So, investigations, replacement, modification or support of anatomy of physiological processes which involves prosthetics, orthotics, supporting or sustaining life, disinfection of medical devices, control of conception. So, many of these aspects like disinfectants for medical devices, control for conception, these are very various areas, like very broad areas in which medical devices come in. This is as per the Ministry of Health and Family Welfare definition, medical devices and cosmetics bill 2022. In vitro diagnostic devices which is a reagent product, calibrator, control material, kit, instrument, apparatus, equipment or system, whether used alone or in combination. They are of intended to use for examination and providing information for medical or diagnostic purposes by means of examination of specimens derived from the human bodies or animals. This is the definition, right, very medical and legal kind of language which is there. So, anything which comes out of the human body, let us say blood sample or a biopsy or any kind of tissue, whatever you use for diagnostic purpose, be it a reagent or control material or a kit, everything comes under in vitro diagnostic devices, right. This is the range of things, again defined by the Ministry of Health and Family Welfare bill. So, why not just have preclinical testing for medical devices? So, ultimately it depends on how the material is performing rather than how biocompatibility is. So, many of us may think that it only depends on biocompatibility of an implant or medical device inside the human body, which refers to how it performs, but that is not always true. It ultimately depends on how this material or a product in whole is performing within the system. Is it causing some adverse reactions? It could be biocompatible, but it may be causing some side effects, or the actual function which it was intended for is not getting met. It may be biocompatible, but let us say an implant is not meeting its purpose of having the load distribution right, or it is not able to bring the spinal column up to its normal form, things like that, spinal implants, right. So, biocompatibility including the preclinical testings still are inadequate as a measurable quantity, right. So, that is why many of the other tests have to be conducted, but the ultimate effectiveness can only be established through clinical trials. So, clinical trials is the ultimate set of steps through which you can actually understand the efficiency or effectiveness of a product or medical device on the human body. So, clinical tests largely depend on two factors: how the host responds to the materials, how the material responds to the host. These both things are very important. Host here is the human specifically or the animal. How the host responds in different forms and shapes, it could be infection, it could be that the function is not met, or things like that. That is how a host responds. How the material responds, it could be that once an implant goes in, it starts wearing out after 6 months due to the body loads which it was not tested for. There could be cases, that is why there needs to be clinical tests, right. It could be that the material is in a little bit of acidic environment and it is the outer surface is getting worn out, or it is breaking due to the different loads, getting ruptured or fractured after a certain point. This is again very much important as a part of the clinical test to assess. Next is typically you have a part of clinical test as an idea of a patient, right. Identify the need for a medical device, you design, select on a material, then you have the final manufacturing and you test it, and you test a material and you also test the product sometimes. So, for example, if there is a certain type of titanium alloy which is used for certain implants, the material was tested first in the animal models or the human body, and then the final product was also tested, right. So, sometimes the material is tested before the fabrication or the device testing or the clinical use, and then you finally test the product, right, as a whole. So, physically you are looking at abrasive properties, adhesive, delamination, corrosion, these are some aspects which you are looking at from a medical device standpoint. From a biological standpoint, you are looking at absorption, enzymatic degradation, calcification, biocompatibility. These are some of the aspects, clinical aspects which you would look at. Okay. Biocompatibility by definition is the ability of a device material to perform within an appropriate host response in a specific situation. Yeah, this is a very clinical definition, it may not give you too much of information. This is as per the DS biocompatibility guidance of use as per the ISO 109931 standard, right. But biocompatibility, like I just explained, it means that how well a material or a device is behaving inside a body. Is it creating some adverse reactions, is it compatible with the human body, right, because it is a foreign material or a foreign device, right. That is what biocompatibility typically means. Sometimes it may create a little bit of adverse reaction, but in an overall scheme of things may fit in really well. So, an implant can have an adverse effect for the first 6 or 10 days, but later on it may be doing really well, right. So, those things have to be considered while assessing biocompatibility. It is not like always an immediate reaction, it is also the overall scheme of things. Also, some other screening tests which are typically considered includes cytotoxicity. These are the terms you will often hear, sensitization, irritation or intracutaneous reactivity, acute systemic toxicity. So, there are a lot of toxic wastes or toxic releases, because whenever an implant or medical device is going inside the body or is in touch with the blood, let us say a syringe is also a medical device, right. It is a surgical or medical device, medical syringe. it is touching the blood. So, it has to, or touching the tissue, it should not have any adverse reactions, otherwise everybody will get a blister or some kind of a infection on their arm and things like that, right. So, there could be toxicity in certain materials like which we use. If the syringe material is made up of certain alloy or a material which releases some toxic waste into the blood, that is not really required or really advisable, right. So, you have to look at the subacute toxicity, genotoxicity. So, there are a lot of toxicity based studies. Implantation effect, hemocompatibility, hemo is like relating to blood, right. Chronic toxicity, carcinogenicity, right. Something should not be carcinogenic to some extent, that is like possibility of causing cancer, right. Reproductive and developmental toxicity, biodegradation, it should not be starting to degrade after it has been implanted or has been in touch with the body after a while. So, if you have a biosensor which is in touch with your body, which is just measuring the sweat levels or the sweat based, like monitoring of your oxygen levels or things like that, so it should not degrade biologically. So, you can very clearly see here a lot of toxicity based tests which are conducted just to make sure that this is properly clinically compliant and biocompatible properly to adhere to the human body. And of course, there is this overall test of how the in the form of clinical trials, how everybody is recovering or this particular device is affecting a person, right. So, this is an example where biocompatibility test based on category or contact duration. This is the FDA's biocompatibility guidance on the use of ISO 109931 accepted in also with the Ministry of Health and Family Welfare Medical Device Act 2017. This act I had introduced during the rules and regulations and standards lecture. So, if you look at the medical device categorization, you are looking at nature of body contact. There are certain devices which are surface devices. This table is very important, this is taken from public information. Contact, if it is like intact skin, mucosal membrane, breached or compromised surface, then you have external communicating like blood path or indirect, then you have a device which is in contact with tissue, bone, dentin for dental implants, like circulating blood, implantable devices like which is in contact with tissue or bone. They have different contact durations, like A is type is limited contact duration less than 24 hours, B is prolonged 24 hours to 30 days, and C is permanent. So, many of the implants or medical devices come within this time frame, right. So, what is below 24 hours? This could be like needles, like certain surgical insertions or absorbable materials, like which absorb within 24 hours, things like that, right. And these have different categories such as like all this biological effect tests like cytotoxicity, sensitization, chronic toxicity, all these metrics are basically followed and tested for these materials and medical devices, very important, just what I mentioned in the previous slide. So, based on this, X is ISO 10993/2009 recommended endpoints for consideration, O is additional FDA recommended endpoints for consideration. Okay. So, few are as per the ISO standards and few are based on FDA's recommendation, and I think most of these recommendations apply in the Indian context as well, right. It is given that note star all X and O's should be addressed in the biological safety evaluation either through the use of existing data, additional endpoint specific testing or rationale for why the endpoint does not require additional testing. So, for example, you are using titanium for a certain type of implant, new implant which you are coming out with. Let us say this titanium alloy has already been tested for all these specific toxicity tests and biological tests, right. Then you can give that justification that this material has been tested, but the new product may have to be tested through few of these tests which are mandatory, right. So, you can bypass a few tests like if they have been already tested on the materials or the baseline product, right. So, these are all clearly laid out what needs to be done as a part of your initial testing for the materials, right. Again reiterating on the classification of the medical devices, because this becomes really important on like we have just talked about the level of interaction in the previous slides, and based on that what all biological tests are must. Just kind of reiterating on that, when you see the intact skin. So, for example, if I am looking at just a insole which is in touch with the skin, you see majority of the tests are not to be conducted. All you have to do is cytotoxicity, sensitization, irritation or intracutaneous reactivity. These are the 3 tests which are recommended, rest are not to be conducted, right. But for blood path or indirect, you have a certain set of tests. For majority of the devices which are in contact with tissue, bone and dentin, majority of the tests have to be done, right. So, this table basically gives a guideline on what tests are needed when the interaction is limited versus interaction is exactly within the body. So, beyond that we need to understand the classes of the device. So, class A is basically low risk, such as thermometers, tongue depressors, right. Class B is low to moderate risk like suction equipment, hypodermic needles, right. Class C moderate to high risk ventilator, bone fixation plate. Class, these are some examples. Class D is high risk like hard valves, AICDs, right. This is as per the Ministry of Family and Welfare Medical Devices Act 2017. We have to look at something while testing this medical devices, which is need versus necessity. So, similar to preclinical testing, not all medical devices need to go through all the stages of clinical trials, right. They have different requirements, depends on the classification of the medical device, and also these classifications are sometimes based on the amount of interaction. So, it kind of goes back to the table for the majority of the parts where you have to look into what all biological tests are important. The need versus necessity also depends upon the ability of the existing data to adequately address the benefit risk profile, claims and the side effects of a medical device. So, it could be that you are coming out with a new dental implant and you had a range of 10 different dental implants which came out of your company, which is the same material and a little bit difference in structure can be observed with the new implant. So, it could be you have a lot of existing data on testing of these implants, the cytotoxicity, like the biocompatibility, the different aspects which you have looked at, and you can use this data as a benchmark and you do not have to conduct these tests over and over again, right. You can now do a few tests only to make sure you are compliant. So, how much is needed, how much is necessary needs to be understood based on one device specifically, and it could be very similar for few other devices, but very different for the other devices. Mostly the implantable devices like class 3 or class 4 needs to go through all stages of the trials. So, irrespective of what have been done previously, if they are completely implantable, they have to go through the all stages of the trial irrespective of what tests have been conducted previously. The ultimate decision lies with the legal authorities, how they want to, whether they want to give some relaxations or whether they want to allow a few bypasses based on previous data or things like that. But typically for majority of the implantable devices you have to go through all these steps. Clinical investigation. What is a clinical investigation? By definition, it is a systematic investigation or study in one or more human subjects undertaken to assess the safety, clinical performance or effectiveness of the product. So, these are the 3 keywords: safety, clinical performance. Clinical performance is whatever performance it was intending to do. Let us say a stent is intending to open a valve, right, then it should do that perfectly. If a spinal implant is supposed to put the spine in order, it is supposed to do that perfectly. So, that clinical performance, if it is not doing that perfectly, if an implant was supposed to bring the spine at a 15-degree to its original structure, and it is somehow making it to bend to 17 degrees, then it is not solving its clinical performance or meeting its clinical performance effectively, right. So, many a times how an implant or a medical device is exactly performing based on their intended use or how the performance expectations were, that is also very important as a part of clinical investigation. It is not always just the safety, right. So, if a hernial mesh, I had taken this example a while back, is placed inside the body, the abdominal region, and it is somehow deviating from its path and not exactly able to being a hammock for the protruding organ, then it is clinically not performing well. It may be biocompatible, it may be safe, but its clinical performance is not good. So, as a part of the clinical investigation, the safety, clinical performance, effectiveness of the product, how effective is it? What is the effectiveness outcome? It should cure a disease or it should mitigate certain problems which were there, right. If there was a device to open a valve, like if there was a stent to open a valve and there is a tissue ingrowth, there is some thrombosis and things like that, then the stent is not performing very well. Or if there is a closure of that artery again, then the stent is not serving its purpose. So, the effectiveness of the product. If the stent is working great for the next 5 years or 10 years, fantastic, it is performing, overperforming from its intended use, right. So, these kind of things have to be considered while doing clinical investigations. So, we are going to end this session here and pick it up from here exactly in the next session, all right. So, thank you all.